It is my great pleasure to invite Jennifer Doudna on the stage. Please put your hands together for Jennifer.
Good.
It's so nice to see you. Yes.
Again, I'm afraid you're someone who needs no introductions whatsoever, and I have about 3 hours worth of questions for you, and we have half an hour to talk. But this is, are you having a good JPM? It's my first question.
Oh, it's fantastic. It's fun this year. I have a nice mix of events that I've been doing, seeing a lot of friends and colleagues, so it's been great.
What do you think is interesting at the moment at JPM? What are people talking about?
I think two questions that everybody's talking about. One is, when is the biotech valley going to turn around? And the other one is what's happening with AI. So it's
well, that's a great topic of fun conversation. So I guess we should talk about that. We know that AI, as we've heard, can accelerate the process of drug development. How do you envision AI specifically helping with evolving CRISPR technology? What do you see as its great power to sort of advance it?
What do I see as AI's power to advance CRISPR technology? Gosh, there's so many ways to answer that question. But I'll just give you a couple of examples. I think that, as we just heard on the previous panel, one of the things that AI is both helpful with, but also in need of, is data and data analysis. And that's something that, we're seeing very much in real time with the CRISPR technology. CRISPR is a great tool for generating data, and for generating data that gives us information about the function of genes or entire pathways. And so I think that, increasingly we're going to see AI being utilized to evaluate those kinds of data sets. And I think that's really very interesting and exciting from a research perspective. And I think going forward, it'll also be informative about the ways that patients are going to respond to drugs. So that's one area. On the more, earlier stage research side of things, something that we're seeing certainly in my laboratory and at our institute, the Innovative Genomics Institute, is that increasingly computational approaches are being utilized to discover new molecules and to figure out what they do. And in fact, I have members of my lab that are here tonight who are kind of driving some of those developments. So I think that's going to also be really very interesting.
And when you say new molecules, do you mean new gene editing molecules?
Yes.
Right.
Yeah.
And so, how's that going to unfold? How does it work? Sorry, I just need a kind of picture in my mind.
Well, maybe I can sort of weave together the pieces of this. So we've heard about AlphaFold. Probably many people here are aware of this wonderful technology that allows you to predict the structure of a protein without actually having to solve a structure and take years sometimes to do that. So that you have that capability combined with a lot of information about the content of genomes. In our case, CRISPR is a technology that came out of bacteria. So we have lots of bacterial genomes that are available and sequences are there. But there's lots of molecules encoded in those genomes that we don't know how to get access to. And so what you can do, it turns out, is that you can take an AlphaFold predicted structure of a molecule whose function and you can use it to go searching through genomes to find molecules that might have a matching structure, but whose primary sequence is extremely divergent or different from anything you've seen before. And that turns out to be incredibly interesting. It's a great way to mine genomes.
So how many sort of gene editing molecules might we discover?
Probably quite a few.
Tens, hundreds, thousands?
Hard to say. I think what I'm thinking about right now is, what are the new capabilities that you want to have in the toolbox?
Okay, so
right?
like fewer off-target effects, maybe, or What do you,
Well, or you want to have modalities that are going to be deliverable into different cell types, right? Of course, you need accuracy, as you said, but you also need potentially interesting different types of chemical capabilities. And I think those are all things maybe out there to be discovered.
well, targeting, I understand. When you say different chemical capabilities, what do you mean?
Well, I'm talking about, different ways that we might imagine manipulating a genetic sequence.
Right,
Whether
so
it's
what, one strand or two strand or?
one strand or two strands, or whether we're changing a letter in the DNA code.
Right.
Are we targeting DNA or RNA molecules? Right. These are all different ways that we can imagine manipulating genetic information.
Okay, so there's a lot of that. Let's talk a little bit about delivery, and that's still kind of like one of the Achilles heels, not just of CRISPR, but of gene editing.
Just in general, or drug delivery in general.
How are we going to make progress with that problem?
Well, again, I'm a big believer in, fundamental science and kind of research driving the discovery of new technologies. And so it's actually a major focus at the Innovative Genomics Institute at Berkeley and UCSF is figuring out how to deliver molecules into cells and to do it in a way we hope that will eventually involve targeted delivery to specific cell types or tissues in the body. This would be incredibly exciting and I think a transformative advance, not just for CRISPR, but really for all kinds of therapies.
And I mean, I've been hearing about envelope delivery vehicles. I mean, in what and how do they work, and how are they in a sort of advantage over sort of lipid nanoparticles, for example?
Well, so you're referring to one type of technology that Jenny Hamilton, who was a former member of our lab, really kind of spearheaded when she was at Berkeley, which was the idea of developing a little package that is based on the way that viruses encapsulate their genetic material, but in this case, using these capsules as a way to encapsulate a protein and a guide RNA, which is really fundamentally what CRISPR is, and to use that little package as a deliverable to address sort of an addressable particle that could be designed to target just one particular cell type. And this is, we're very excited about the potential of this to enable targeted genome editing in the body, which is not something that's happening yet, but I think we're on the verge of seeing that become a real capability.
Like loads of questions. So the first one would be, does it generate an immune response?
I think it could, but one of the great things about CRISPR is that in many cases, it looks like we're going to be able to use this as a one and done type of a therapy.
And then can you get it past the liver?
How do I get it past?
The liver.
How do I get it past the liver? Well, this is where the addressable factor comes in. Because if you can target a particular cell type, and that's done by having these little particles recognize molecules that are just presented on one particular type of cell and not others, then you can, in principle, have those particles going directly to one cell type in the body and not to the liver.
Do we have enough unique biomarkers to target all the cells we want to target?
Not today, but that's where we need more discovery.
Wow. Okay. So manufacturing. Can AI help us with manufacturing CRISPR molecules?
Yeah, I think... I'm sure it can. I think, again, my feeling is that we're still going to really need to develop the kinds of data sets that will be appropriate for training. And, we heard about that in the previous panel. I'm sort of clearly on the side of, we need more data, we need more good data sets. And I think Alex Gorski actually made a very interesting comment that I think about a lot, which is that I think there would be real value in being able to aggregate data sets. I'm not sure how to do that from a regulatory or sociological perspective, but scientifically there would be incredible value, because you could imagine that if you want to be able to manufacture molecules more efficiently, you need to know a lot of things about not only their chemical and biological properties, but you need to know things about, just very practical aspects of how to produce them efficiently and which ones are going to be most effective in which types of patients. And so if you could aggregate those types of data together, I think it would be very powerful.
So what sorts of data were you talking about specifically? Patient data, manufacturing data?
All of the above.
All of the above.
Yeah.
And there's nothing kind of like it. There's no sort of, are there any sort of precedents for this kind of thing? I mean,
I think not. And, one of the things we're discovering, just, taking a step back from that and looking at what we're actually doing in our lab and at our institute, which is more on the, really preclinical level, or just, doing research, is that we find that, yes, there are lots of data sets that are available for different kinds of biological experiments, but for the most part, they are not aggregated. They're not in formats that you can easily combine. And so one of my colleagues, who's here tonight, in fact, has been just working on doing just that, sort of looking at what's out there and available in the public domain for a particular type of data, and just spending time, it sounds kind of boring, but just, putting those data together and curating them, so that you can then use that as the basis for training models. And I think we're getting to do more of that.
You highlighted the potential of CRISPR to address issues like climate change and carbon capture in plants and things like that. What do you think are the most promising areas for CRISPR, sort of beyond humans, basically?
Well, I guess I like to think of human health in a very comprehensive way. Our health obviously depends on our bodies, but it also depends on the food that we eat, the air we breathe, the environment we're living in, other organisms in our environment. And what's really exciting about CRISPR, and it was clear from really the very beginning with this technology, is that it is a platform in the sense, right, that it's really cross-cutting. And it allows us to manipulate organisms of any type. So our institute was set up with the idea that we're interested in having global impact with the technology. We want to make it widely available, and we want to have, we want to be able to solve some of the biggest problems. And clearly climate change is one that is, really an existential threat. And how do we do that? When we first started talking about using CRISPR for climate, I had a lot of scratching heads and puzzled looks, but if you think about it, there are some really interesting and really concrete ways to do this. And the one that we're probably the farthest along with at the Innovative Genomics Institute is the approach of manipulating the microbiome. And we've got a project that involves scientists at the University of California, Davis, who just signed on as one of our IGI partners, where we can actually go into the microbiome of the cow rumen and manipulate those bugs so they no longer produce methane.
You're talking about farting, basically.
Or burping.
Oh, of course, yeah.
And the amazing thing is it works, and so what the path we're on right now is, how do we deliver it? How do we scale it? And how do we make it go global? And so these are problems that a lot of our scientists, a lot of them are early in their career, they're very excited, they want to contribute to this type of a very big global challenge. They get very excited about this, and they want to come in and innovate to solve this.
I mean, what kind of impact could that have, do you think?
Well, I was shocked to discover that animals and farming contributes a huge amount of the greenhouse gas emissions that happen around the world annually, especially with methane. If we could make farming effectively carbon neutral, I mean, that would have an enormous impact on reducing the release of greenhouse gases every year. So this is something that I think there's a very tangible, very clear goal of that research, is to achieve that.
I mean, do you really think we can engineer plants to do carbon capture maybe more efficiently than the other?
No doubt.
So why is that such a certainty? I mean, I'm just interested. I'm not.
Well, here
I'm just saying. Well, what makes you know that?
What makes me know that? Well, here I'm thinking of the work of my now late colleague Joanne Chory down at the Salk Institute, and she led a group there that's continuing to develop plants that have extended root systems where you can ensure that, there's going to be long-term carbon capture physically in the soil with these root systems, and using CRISPR to do that. So, that wasn't a fantasy. It wasn't maybe we can do it. They're actually doing it.
And could we do it at a sort of meaningful scale, do you think?
I think the answer is yes. It's going to require, but it will absolutely require scientists, going beyond just publishing the next academic paper, right? We have to be working with farmers. We have to be working with communities globally. We do this at the IGI, is really reaching out to people in other countries to help them understand how this technology could really impact what they do.
I mean, I'm interested. You published your science paper, 2012, right? Was there like a kind of oh shit moment when you kind of like really realized that this was like fucking huge, excuse my language, like you're like, oh shit, what have I invented kind of moment? And what was that? Like, what were you imagining?
Well, I've told this story before, but for me, I can remember there's sort of two moments. One was in the lab, and one was at my house. So the moment in the lab was working with Martin Yunek, who was the scientist in my lab, who was actually doing these early CRISPR experiments, and being in the lab with him when he performed the experiment that showed us that CRISPR is a programmable enzyme, and that we'd figured out how to program it by changing its molecular guide, which is a molecule of RNA, to direct it to different DNA sequences. And literally being in the lab and leaning over the light box in the lab and looking at the result and saying, oh my gosh, this is really, really cool. And so there was that moment. And then there was the moment that it was probably, not too long after that I was at home, and my son was about 6 years old at the time. I was cooking dinner for him, and I was thinking about this, and I was thinking about the data that Martin was generating in the lab, and I just kind of burst out laughing in my kitchen. My son said, "Why are you laughing, Mom?" And I said, "Because bacteria have this incredibly cool molecule. You have to hear about this."
But I mean, did it take time to sort of realize just how impactful and how many kind of areas of biology you would end up? I mean, it was research, it's human biology, it's plants, it's agriculture, it's bioterrorism. I mean, there's just like sorry, but I mean,
throw that in there.
Well, but it's like everything, right?
Well, I think the thing about the CRISPR technology is that, it was clear from the very beginning that this was going to be a great way to manipulate DNA sequences.
Right.
But of course, it was hard to imagine exactly where it would go and where it would have the most impact. For example, could I have imagined back then that within 10 years, we would have an approved therapy for sickle cell disease with the exact same molecule that Martin Yunek was working on in the lab? No, that would have blown my mind, but that's what happened.
I know, it was incredible how quickly it got into the clinic. And I mean, I've watched gene therapy for years, and it's like just CRISPR sort of was on the starting line behind gene therapy, and it's sort of like gone past it. Anyway, so right, where was I going to go with this? What keeps you up at night, if anything still does?
Oh, sure. No, things keep you up at night. Look, I really want this technology to solve real problems, and I want it to do it on a scale that's meaningful around the world, and that's a tall order. And how do I do that? So I ask myself frequently, where can I be having the most impact with my time? And I, continue every day to come to the conclusion that it's by doing exactly what I'm doing right now, which is, working at one of the world's best public universities with some of the smartest students and postdocs and colleagues that I could imagine, and helping the ideas that are coming out of that research effort and at our institute, which is part of that, to see to have the opportunity to scale those efforts through entrepreneurial programs that we have and through helping new companies to spin out. So for example, at the IGI, I think we've now spun out close to 30 startup companies over the last several years. And those companies are working in a number of different areas. A lot of them are in healthcare, but some of them increasingly are in synthetic biology or even in some of these more climate and ag-related areas. So I'm very excited about that. I think those entrepreneurs, a lot of them are former lab trainees of mine and of my colleagues. And these are people who are going to change the world. They're the next generation. We're going to make the next set of breakthroughs and scale them.
When we're here talking sort of in a venue about sort of AI and drug development, and are there any, and you've thought a lot about the ethics of CRISPR and things like that. Do you think that there are any lessons that you've learned about kind of communicating and learning about the issues surrounding your technology that perhaps the AI community could take from you about how to handle sort of ethical issues or concerns or something like that?
Well, I think one of the things that I feel strongly about is that we have to have first of all, we have to acknowledge that there was a challenge there,
right?
And that there needs to be discussion, there needs to be transparency around it. I really admire the work of Fei-Fei Li at Stanford. You know, she's one of the people in the AI space who I think has really been leading the effort to think about the ethical challenges with AI. And you know, we've had a number of conversations about the parallels with, you know, what's happening with CRISPR and the ethical challenges there. I don't think there's a one-size-fits-all solution, but I do think that having scientists, people who are actually involved in the technology development, be part of that conversation is essential. And it's a little bit uncomfortable for a lot of scientists, you know, at least in my experience, you know, and I'm talking about myself too, you know, it's not something that we're necessarily trained to talk about or think about, but it's critical that we get out there because, you know, especially in this day and age when there's a lot of misinformation out there, there needs to be a voice or voices of fact and reason and people who are saying, look, this is what's really going on and help those of us that are not experts to understand it.
And you said that there are parallels, you think, between what you're doing and what Fei-Fei Li is doing, right? Do you have some sort of examples, perhaps, or something?
Well, I think just, you know, being willing to get out there publicly and speak about these issues is very important.
And, you know, is there a sort of tension not only between, you know, the kind of wanting to sort of talk about your research, but also most scientists just want to get on with their research. They don't,
of course,
right, want to talk about. So how do you get people to do that?
Well, I think first of all, not everybody will want to, and that's okay. You know, that's okay. But some people do, and I think just looking for those that want to rise to that challenge is important.
What in the next year, 2 years, what do you think is going to be most exciting? What's happening on the horizon? What's 2025 going to bring for CRISPR and for your world?
I think we're going to continue to see the technology advancing very quickly. I'm excited about opportunities that are going to come about from the convergence of technologies, including, we talked about some of them already, but, you know, certainly the ability to deliver molecules to cells in vivo, combined with the most advanced ways of manipulating genomes, and then perhaps adding on a layer of AI guidance for what kinds of genes we should be editing and what effects they're going to have on patients. I think that we're going to really increasingly see that kind of convergence having an impact. There are a number of companies that are already pretty far along in terms of clinical development of different applications of CRISPR. I'm expecting that over the next 18 to 24 months, we're going to see those starting to go to the FDA for approvals, which will be great. And then I think that there's a whole cohort of newer technologies that are coming along in startup companies that are looking at ways to use CRISPR creatively, whether it's to manipulate RNA molecules or to make other kinds of targeted changes in DNA, or even make multiplexed changes in DNA that I think will be exciting. So I'm very bullish on the future.
So the multiplex changes, I mean, you're talking there presumably about being able to address more common diseases by doing, is that kind of the goal, right?
That's right. Ultimately, yeah, being able to do that would be important. Yeah.
But then sort of the challenge there, isn't it, that these technologies are really expensive. And so how do we kind of get, if we want to, I don't know, gene edit for cardiovascular disease or obesity or whatever it is, how do we transform this technology, this very kind of expensive, difficult to deliver technology, into something that is much more affordable and accessible?
Right. Well, to answer that, I guess I would like to refer to a program that my colleague at Innovative Genomics Institute, Fyodor Urnov, is running.
I know Fyodor.
You know Fyodor, yeah. Most people know, if you've heard of Fyodor, he's a wonderful guy, and he has spearheaded an effort to really ask that question, but actually in the context of rare disease. And before you say, well, wait a minute, I don't want to just talk about 3 or 4 people. If you put together all the different rare diseases, it's actually a large number of people that are affected. And so, the sort of the double-edged sword with CRISPR is that on the one hand, it's a programmable technology that could, in principle, impact all of those patients. But in practice, it's very hard today to actually do that because it's hard to, we just don't really have a regulatory framework that would enable this. So, what do we do? How do we deal with this? And Fyodor's approach, and I think it's really very exciting, is to say, well, let's first of all ask ourselves, what needs to change in the way that we currently make and test drugs so that this could be a possibility, and then working with the regulators to, really engage and think about how we could work together to change that clinical pipeline for testing, and partnering with, in our case, Danaher, which is a conglomerate company that brings together many manufacturing capabilities that, academic labs and nonprofits, otherwise might not have access to, so that we can actually make the molecules that we need for these rare diseases. And we hope that, we can really start to forge a pathway that we would love others and companies, of course, to be able to follow, that will open the door to, using CRISPR in many more settings.
The MHRA in Britain has started to think about individualized medicines, and I know that the FDA has started to think about it too.
Yeah.
I mean, do you have an idea about how long it's going to take before we get to a position of being able to say, give individualized medicines? It could take a while. And how much of a priority it is?
Well, it's certainly a priority for us and for me personally. And how long will it take? Well, I think, our plan at the IGI is, and working with our clinical partners at UCSF, we would really like to have at least one clinical program launched in the next 5 years and have that
individualized medicine.
Correct, but with the idea that CRISPR becomes designated as a platform where you can easily change the guide molecule that directs it to a particular sequence, but have it be under one testing umbrella.
And if you're talking about rare diseases and genetic editing, you're presumably talking about children, right?
In many cases, yes.
Yeah. And so I've talked a lot to the mother of Mila, Julia, about Mila Sen and how that was developed. And her argument, her point of view is that there's not enough risk taking, and that basically, if you're in oncology, right, and you have a child at the end of their life, the oncologists are really kind of quite willing to take risks with, well, we'll try this, we'll try that, on the off chance that, you'll find something therapeutically beneficial. But, her view is that, when it comes to treating rare diseases in children, that there's much more caution, and there isn't the same tolerance for risk. And, that's a kind of cultural thing as well, and that's hard to change. Do I mean, you're nodding, so you obviously see that. But I mean, is there any way we can kind of get around this? Because obviously Julia says, well, when you have a child who is going to die, and that, you have the opportunity to do an individualized medicine that could save their life, but they're going to die anyway, and horribly. Why isn't it easier to take that risk? Why do you have to go through this sort of 2-year development timetable, customer? There's a whole pathway for it, but it's quite difficult and challenging. So, yeah. I mean, how do you convince people that really this is ethically right and moral to take these risks?
Well, which people are you wanting to convince?
Well, the regulators, because they have they're very risk averse.
Right. Well, again, I think it is going to require it's going to require a lot of handholding and a lot of discussions and probably engaging with these patients themselves, right, to advocate for that perspective.
Right. Anyway, so cost. So how do we get to the cost piece? Did we get to that question? I don't think we did.
We didn't, but and I see we're close to time. But I'll just make one comment here, and that is that Melinda Kliegman, who until very recently was our public impact director at the IGI, ran an 18-month program in which she really worked with people who are both clinical and science experts in genome editing, but also people who came in with a regulatory perspective. And the result of that analysis of, how do we reduce the cost of genetic therapies? How do we make them more widely available? Is an article that ended up getting published in Nature magazine recently that I would refer you to if you're interested, that really does lay out some very specific ways that we can work together to reduce the cost. And quite just in summary, it comes down to, continuing to advance technologies like in vivo delivery methods, as we discussed, continuing to think about how we're going to reduce the actual cost of manufacturing these molecules, and then working with regulators to streamline the approval, testing and approval process. And that's going to be really key, I think, those three things to helping to bring down the costs.
Well, look, this has been absolutely fascinating, and you're incredibly quick thinker and responder. You've kept me on my feet for half an hour.
I could say the same about you, Natasha.
So look, my job now is really to sort of close this session, but I'd just like to ask the audience, please could you thank Jennifer for being such an incredible...
Sure. Thank you.
Thank you very much.